This computer. Okay, now we are recording. Hello, Peter. It's very lovely to talk to you. And my first question will be to introduce yourself and Agena, just to give a bit of general view of what we're going to hear about today.
Very good. Well, my name is Pete Dansky, and I'm the CEO of Agena Bioscience. My background, I've been an executive and spent my career in the life sciences tools business. Actually, I started at school as a chemical engineer, and then when I graduated, I realized, gee, maybe I should have taken some biology because I'm kind of interested in the life sciences. So I just took that tack pretty early on, and it worked for a large number of different companies. Actually got involved in the Human Genome Project with one of the early DNA sequencing programs, which is when I got involved in genomics around circa 2000 or so. And since I've been involved in different businesses in the genomics kind of area, and particularly passionate about life sciences as an industry that is able to really make a difference in people's lives. So as far as Agena goes, Agena was formed about a little more than 6 years ago. It was a technology that was spun out from a previous public company called Sequenome. And the technology is our mass array platform. I'm sure we'll spend some more time talking about that in this interview. But really the purpose of the company and our history has been to take this platform that had been well utilized in kind of research applications and move it into the clinical diagnostic space.
Thank you. Have you ever considered a career in academia, and how do you see, like you mentioned that the tools are very important for the research, and how do you see the synergy or competition of academia and business in driving innovation?
Well, first of all, it's an easy no. It was never an interest for me to personally pursue an academic career. I think both because, I mean, I did spend some time in school doing some research and as part of a graduate program, but I'm interested in a little more immediate gratification, I guess, call it patience, but also found that where I really thrived was working with teams of people to make products and to deliver products to the marketplace so that so researchers and other clinicians could use them. So very early on, I established my own interest, but I've always seen a very strong synergy actually, and probably the easiest way of differentiating is that in academia there's more our true research where questions can be asked and answered and pursued with a long view and maybe a more transformative view. And I see, companies more well positioned to do the development or the D part is to take those ideas and inventions and turn them into real innovations, where an innovation is not just a new idea, but an idea that is brought to purpose to make a difference in people's lives and in the marketplace. So I see a lot of synergies, and have had experience working in a variety of collaborations, and we at Agena we work hard to interact with opinion leaders in the different application areas we're pursuing. The so-called key opinion leaders are those that are more on the research side, whether it's academic research or in our case more clinical research. There's still a very strong partnership and collaboration that leads to publications and establishes the proof really of reference. That's both important as far as the data that gets established, but also the influencers, the human beings who are in academia on the front end, who are the influencers, the people that others listen to. So I think it's really important, and it's part of a big part of our scientific affairs strategy to make sure those partnerships are working well.
Thank you. You already mentioned your way to Agena. So it was if I'm not mistaken, mid 2014, where indeed mass array technology that you've mentioned was mostly used in scientific research without obtaining the market for clinical diagnostics. On the other hand, NGS technologies have already firmly gained the sequencing market and continued reducing costs and improving accuracy. What were the risks back then, and how you were calculating them, how you were seeing them?
Yeah, well, thank you. It's a great question because, as I said, the initial premise of establishing this company was that the technology we have was well suited for the clinical space, and we can talk more about that. But this platform is, while it had been used in research, we saw it as being really ideally suited as a clinical platform in terms of its data quality, whether it's accuracy, precision, the throughput, the flexibility, ease of use, low cost, really well suited for diagnostics, but it just had not been really applied there in a significant way. And of course, there's always risks, right? So, 6 years ago, let's say the two biggest risks that kind of hung over this business and ones like it were, as NGS. What is the trajectory of next generation sequencing in the clinic, and how might that play out, as well as a regulatory risks, particularly in the US and also many European countries where, and it is the case that much of our business is in enabling clinical laboratories to develop their own tests, so-called lab developed tests, or LDTs, or sometimes called home brew, and that's that's the vast majority of our business has been and is in that kind of open platform approach. And there was always the uncertainty as to whether that avenue would stay open or whether regulators would close it down more. So with regards to both of those risks, I would say, with regards to next generation sequencing, it's a very powerful technology, obviously, and there's massive, multi-billions of dollars of investment in very large franchises. But actually, the promise has not, the results have not met the promise from a clinical perspective. 6 years later, if I take a lens on NGS in the clinic, I don't think it's moved forward significantly even since then. In terms of complexity, the ease of use, the cost, and frankly, the relevance, right? Because I think, and this is probably the most compelling part, is that, when you're in the research world, right, anything is possible. And the notion of, thousands of markers or the whole genome, right? And somehow we can magically, take this data and interpret everything. And, I mentioned in my career, I was involved in serving the Human Genome Project. And that was very kind of similar concept at the time, that, well, if we just map the genome, then everything is going to be elucidated. And of course, it's just a starting point. And, true advancement is slower, but, in the clinical space, it's particularly true because, a clinician needs to do something with the data. Otherwise, the result is worthless, right? And so, in a sequencing application, so many what are called variants of unknown significance, there's something there, but the doctor doesn't know what to do with it, or the patient, that's a vexing problem. And even without that, it's just, useless information beyond what is really what we consider actionable. And so, in short, on NGS, we continue to see, we've really seen little inroad in terms of the true value proposition getting stronger relative to what we offer. And I'm confident, long term, that there's a place for this kind of very targeted testing for the clinic where, 25, 20, 40, 60, even 100 variants, that's an incredibly complicated clinical test, right? 5 or 10 is, a lot, right? And thousands, I think, is a distant fantasy, frankly. I think from a regulatory perspective, of course, that's played out well, and we've been very strategic in markets like the US, where laboratories are enabled to develop their own tests, and that's continued to be verified and expanded, and particularly in in the throes of the pandemic that we're in, we're seeing, you know, that happening real time. And so we benefit from that as a provider to that space. But also in certain markets, and certainly Russia is a case, and China has been a significant case in our kind of, from the start of our business, you know, approved diagnostics are really required in certain countries in order to really sell into medical institutions and have them used. And so, you know, we've tended to take more of a partnership approach for the in vitro diagnostics, and leverage our technology into other partners who can kind of take it the rest of the way through approval.
If we go back to 2014, what immediate actions did you take back then? What was the development plan that you had when you just joined the company, and how did it work out, or how did it change?
Well, of course, it worked out incredibly well. I wouldn't say otherwise, but I can just tell you the story. I mean, and this is, you know, if you ask me about any business or situation that I stepped into, it would kind of follow this kind of paradigm, you know, team first, and very much team first. And so, you know, I established a leadership team, and that included, you know, both, you know, people who had come with the acquired business in various leadership positions, and then also new roles. And remember, what we acquired was a business, not a company, right? So we set up a full operating company, and many aspects of the previous company stayed with the initial company, and so we had a lot of elements to build. So, you know, building the leadership team was number one, but then the culture, right? I'm a big believer in values-based leadership. We've established a values system that I've used before, and has really resonated at Agena, and is easy to remember. We have 4 T's. There's teamwork, transparency, transformation, and tenacity. And those are really guiding principles for the company and how we operate, you know, how I select leaders, how we interact together, and how we build, you know, cohesion and effectiveness within the company. From that's kind of the preparative steps. The first thing I did was go visit a lot of customers. So in the first 3 months, you know, every region of the world, go visit, because it wasn't ongoing business, of course. And we did have, you know, maybe somewhere between 10 and 20%. I think it's a little hard to look historically before our records, but less than 20% of the business was already operating in clinical laboratories. So this wasn't a completely new concept, which also was a risk reducer, obviously. But went out and met with customers around the world, and also potential partners, particularly in China, where I was already considering that strategy, and as well as the company's employees, right, who are also, we acquired employees all around the world, and so built that knowledge set and relationships with top customers and employees around the world. And then as a leadership team, we came back and really put together, you know, our focus strategy and our product roadmap into a variety of areas that you see us playing now in you know, both on our platform in terms of making it more clinically relevant and robust for clinical laboratory workflows, and then a range of applications that just made sense for us to pursue, including oncology and pharmacogenetics and others. I guess the last point I would make, and I know we're going to get into this, I imagine we're going to get into this, and you know, every region's a little different, right? And so, you know, in the case of the US, particularly in that time, pharmacogenetics was really expanding. It had become very popular, was you know, many lab developed tests, competing platforms, and we believed that we had the best solution. And so we put a big focus commercially and also in development on establishing our position in the pharmacogenomics space, which we did do very successfully. But then as I mentioned, in China was the other major market, completely different strategy. So the first steps there were setting up a wholly owned subsidiary and building a leadership and business team. When we started, there were just several people operating in China for the company without a subsidiary, sort of what's called a representative office. So we established a facility, a formal operation there, an entity, and we now have 35 people in China with a very robust business. And the first steps there strategically were to establish partnerships, as I mentioned, for diagnostic development, and those we have many of those now, which are underway. And all that was really initiated in the first 6 months, to get that started.
You already touched a little bit this topic about the teamwork. So can you elaborate a bit more on that? How responsibility is divided between people in your company? Who generates ideas about which segment should be developed? How is the decision-making process organized?
Sure. Yeah. And it goes to some of my leadership style. I mean, first of all, as I mentioned, team. It's a lot of team dynamic. Functional leaders, though, they own their areas. Team doesn't mean, first of all, that everyone's involved in every decision, and nor does it mean that it's a democracy, right? I mean, my approach is what I like to call consultative. I want to make sure to hear all views, especially minority views, and that's where the transparency is so important, to be receptive to all the ideas. And so ideas come from everywhere, but certainly functional leaders in their areas are given broad accountability and authority for their areas of the business and their strategy. I operate my senior leadership team as much as possible like a board of directors. So there's functional representatives, but when it comes to the big issues of the company, we discuss them from an even kind of perspective across the team, and get so that we get hopefully the best decisions from that perspective. We also, at the next level of the company, I've set up an extended leadership team. It's the top 10% or so of employees, or top is a variety of employees who are involved in particularly involved in either a lot of cross collaborative activity in the company or more strategic kind of roles, and the most senior people in the company. And that we engage regularly as a group to really get more input for decision making, but then really be building a deep alignment to ensure that everybody understands where we're going. In terms of where we're going, marketing is really the responsible point for product direction. Marketing should be collating a view of where we're going, and they do, and in R&D on more about how we're going to get there. But of course, ideas come from everywhere. They come from R&D. They come from the whole team, and especially they come from customers. Right? And with regards to that, when we think about our platform itself, our core technology, we know more about that than anybody, and so it tends to be the case. And most of what you're looking to do are pretty obvious improvements. Boy, I'd like it to be better or faster or cheaper or easier, right? And so ideas around the platform tend to come from within the company, and particularly from within the R&D organization, in terms of either improvements to our instrumentation to improve its workflow, or new chemistry approaches that can enable different applications. But the content tends to come from customers. And we have and we talked earlier about key opinion leaders in academia. More generally, we interact with our customers extensively on what content they want to deploy on our platform, because it's a very customizable platform. And so every customer can essentially have the content that they want. So we have that flexibility, but then we keep sensing of where customers are going, because you see the trends, and you can see, oh, there's a coalescence of interest around certain sets of content, and that has the potential of becoming a more standard product. And we have our scientific affairs group is especially charted with kind of tapping into that customer interest and working with marketing to align on new product ideas that are coming from customers.
I see. You talked already a bit about your leadership style. What would you consider your first success as Agena's leader?
Difficult to pick. There's a lot. I mean, I think if I step back now, because it's interesting now, you're asking questions about 6 years ago, and it's different. A year after you start versus 5 years after that. But when I step back and look at it, the success with which we pivoted to molecular diagnostics was quite remarkable, right? And I pride myself on that, though it's not a stated value. Maybe I need to come up with a fifth T value word for adaptability. We pivot very quickly and effectively, and that's somewhat in the transformation notion, right? We as I said, we had less than 20% of the business in the clinical space when we started. We're over 90% this year, right? And some of that is that the legacy older research businesses declined and people have stopped doing that, but we've had incredibly successful growth in the clinical space. And so fundamentally our strategy has paid off, and that's a long-term measure of our success and defines the future. But it did happen very quickly, especially in the US. And I think in the US, it was the early traction around pharmacogenomics, which is now expanding all around the world, right? So several years later, we now have significant uptake and interest in pharmacogenomics across many European countries, as well as we have multiple partners in China developing in vitro diagnostics for different pharmacogenomics applications, and it continues to be very, very, very dynamic and growing within the US as well. So I'd say with regards to the US, it was pharmacogenomics. I mentioned our partners in China. I think that the measure of success that I see in China is in the degree of interest in the brand and the product. We're actually very well known there, and we have. And I saw frankly, even before Agena started, I think the predecessor company had done a pretty good job because the benefits of our platform, they are particularly relevant in China. Very high throughput, very high volumes of testing. Cost is very important, and the technologists who can run these systems don't always have the sophistication that some other countries' laboratories may have. And so ease of use is extremely important there. And so our brand has been, extremely well appreciated. And, I saw, early uptick. And now when I look back, I think that the degree, the speed and intensity and I personally spent significant time in China, meeting partners, building relationships, building our team, and that was an investment, early investment that's paid off very well.
Thank you. So now we can move from 6 years ago to now.
Mm-hmm.
To mainly the technologies. Speaking of such and its possible applications in the areas where mass area wins over other tools. So it's, as you mentioned, pharmacogenomics, pharmacogenetics, tumor typing. It is competing with NGS and droplet digital PCR, and they are developing two. Can it happen that the sensitivity threshold of NGS will become even lower than that of your system, and the price will become comparable? Are there any preparations for this case? How are you considering this kind of turn?
Yeah, it's not exactly how I see it. I think that, because firstly, you do have to talk about, kind of a line on different applications and what matters. So first of all, maybe just to try to unpack this a little bit, because when you bring up the point of sensitivity, sensitivity is important in certain oncology testing, particularly in liquid biopsy, and so we should talk about that. Other application areas like pharmacogenomics or hereditary disease testing and many others are it's really not a sensitivity issue. There's plenty of sample. It's just accuracy and consistency of results. So, but since you asked about sensitivity, my view is in the area of liquid biopsy, there is a level of sensitivity that is clinically relevant, below which actually it's diminishing return, right? So, we're already operating at a level, where, what we call the minor allele frequency, or the sensitivity of able to, how many straws of hay are there with the needle in the haystack, 1 in 1,000 kind of level that we're getting down to now is kind of the threshold of clinical utility. Because the reality of it is, even if you could, let's say 1 in 1,000,000, no clinician is going to act on that because it's just too spurious. And so I think there's pretty good alignment amongst opinion leaders that the kind of sensitivity level we're at right now is relevant. And that's not really what is going to affect the adoption of liquid biopsy tests. All right, it's about proven clinical utility and changes of behavior of clinicians who are used to, for example, doing diagnostic imaging, to add in a molecular test at the same time. And that's where the evolution is happening. I think that also, one needs to be very wary of arguments for next generation sequencing being more sensitive, because sequencing can be as sensitive as you like it to be. You just need to sequence more. But it comes at a cost, but more importantly, it comes at a cost not just of financial cost, but it comes at a cost of specificity. Because the deeper you dig, the more you find, and some of what you find are false positives and some are false negatives. And it becomes an incredibly complicated bioinformatic challenge to discriminate that. So we really don't worry about sensitivity. I guess that's the first point. And you mentioned digital droplet, which also is notoriously very sensitive. But here's the issue, in the case of digital droplet and again, I want to talk about cancer monitoring, because I think that's really where the field opportunity is and where we fit. Monitoring a patient to see how if their therapy is working or not, if there's residual disease, if the cancer is coming back, if it's mutating as it's evolving under therapy, that can really inform what the next therapy is. In the case of digital droplet, it's a very low density application, right? And all the clinical guidelines, call for, there are now dozens of relevant markers, for example, in lung cancer or colon cancer and kind of every area. It's not 1 or 2, right? It's, our panels have 50 to 100 markers on them, which is a perfect kind of mid-density application space where we play. For digital droplet, you'd need to take, a full blood draw for, every few markers that you want to measure. It becomes completely impractical unless you just want to monitor, for one particular marker. So we don't see a lot of longevity actually in true clinical applications. And then sequencing, it kind of falls on the other side, right? And actually, again, too much content at too much of a price and a complexity and to run the tests. And it's one thing to do an initial diagnosis on a patient, and those tests are out there today. But actually, in the U.S., the agencies have determined that NGS can only be reimbursed once on a patient, first test. So if you're going to follow up and monitor, they won't reimburse that. And that's where a mass array technology we see really fitting in at the perfect sensitivity level with the density of markers that you want to monitor, but at a price and a turnaround time that actually allows that monitoring to take place. So, and again, if I go back to the history as being a reference for the future, sure, you can say, well, if theoretically any technology showed up that was a black box that was perfect, it could be better. But those are all just theoretical ideas. And when I look at how sequencing has advanced, it's only really in the big laboratory companies that have massive computer infrastructure where they're able to discriminate, effectively these NGS tests, and it's still I think a very big challenge in the actual operating clinical laboratories, and we believe in a more distributed model.
Indeed, my next question would be more like about distribution, because molecular diagnostics is actively developing these approaches to the point of care testing, and your hardware being a little bit mobile, but also it requires large amounts of analysis to recoup the investment. How would you combine this, movement of market and your technology?
Right. Well, I think the question of point of care versus the central laboratory is an ongoing dynamic, right? This is not a new trend, and both are relevant, and I think both will continue to be relevant. I actually don't see that more diagnostics volume are going to point of care. There are more tests being developed, but the real question is what's the need? What's the clinical need of that test, right? So in the area of many infectious diseases, and we'll put COVID aside for a moment, because when you reach a pandemic level is a different kind of scenario. But much of in many infectious diseases are tested on patients who show up in a hospital, right? They show up at the doctor's office very sick. They show up in a hospital, and they need an immediate test to determine, do they have a virus or a bacteria, or if so, which antibiotic to give, or is it a viral treatment to give, or so on. And that's the kind of test that needs to happen very quickly. And that's where, a point of care test makes a lot of sense. In many of the applications that Agena is involved with, and they're all growing, right, dramatically, whether it's pharmacogenetics, even cancer profiling. I mean, sure, you want to know quickly, a day or two, that's fast, but you don't need to know in an hour, right? And it's not, other than peace of mind, there's no real clinical benefit to that. And there are significant benefits economically to the efficiencies of operating in a central laboratory. And so that's why actually, if you just look at the market today, and even though there are many point of care tests, the vast majority of clinical tests are still done in large laboratories. And I actually don't see that changing. I see, certain tests will move closer, either point of care or closer to patient. Many others will continue to be done in high throughput in laboratories that we serve. And again, as is often the case with our technology and approach, it can be nearer to patient than, for example, sending out to one national laboratory that does all the testing that needs to be sent out and the results back. It could be done in a central lab within a hospital system, for example, or a local laboratory. So the degree of where we could be a little closer to patient than, let's say, a big factory. But again, I see, I've never considered that, much of a threat in terms of the longer term viability of diagnostics. And that's not just for Agena, right? I think that central laboratories will continue to persist. And the last point I would say is, it's true kind of in every industry, right? You have natural drivers toward consolidation for efficiency. And the same is true in clinical diagnostic laboratories. So I expect that we will see, for central laboratories, there'll be more aggregation, fewer laboratories doing higher volume of testing. As an example, great example of that's in the United Kingdom, where they have moved from I think 11 or 12 clinical genetics laboratory sites to 7 across the country as part of the National Health Service. That's a whole sort of restructuring to create more efficiency across the country. And I think we see that trend happening, generally around the world, where larger labs are getting larger, or big commercial lab companies are buying smaller ones, and this is the way that industries evolve. So high throughput platform like mass array should have a strong future in that dynamic.
You've mentioned already liquid biopsy, as one of the possible point of growth. How do you see its future? How do you plan to implement it, like being quite well developed, but into clinical practice, into like talking clinicians into moving into that direction?
Yeah, and as I said earlier, I think that I'm not in a position to provide a prognosis of how big this market will be and when it'll happen at significant levels. But all predictions are that it'll be huge, right? And you can find many reports and analysts and view on that because it's so compelling, right? I mean, it just makes so much sense. But you have to really break it down into different areas of application, and particularly whether you're talking about before a diagnosis or after a diagnosis, right? So, and we're focused more on the opportunity after a diagnosis. We think it's well suited to our platform, and it's more immediate, right? It's more relevant immediately. And this is the idea of doing therapy monitoring after initial diagnosis, wherever the initial diagnosis may have been done profiling on a mass array platform, or it may be a sequencer, or maybe any other technology. But once the patient is diagnosed and they're put on a medication, which would be let's say a targeted therapy, you want to know if the therapy is working, both with regards to is the tumor still there or not? Is it coming back? And if it does come back, is it mutating? And that's really important in both lung and colon cancer, where there are second and even third line therapies, because it's known that when the tumor is attacked with a given targeted therapy, that attacks essentially one mutation, that other mutations may emerge and then create another growth path. And so you have to keep kind of knocking it down. So that kind of monitoring testing is, we think, where the opportunity is. The content is already quite well understood. It's the same content that you test a solid tumor on, but you can do it in a monitoring scenario without having to do a rebiopsy. It's a very powerful proposition. The other, how soon that's going to take off, at what level, I don't know. What I can tell you is that we're prepared for it, right? So, our effort has been, again, working with key opinion leaders. There are now a very strong set of publications and collaborations and consortia who we've been involved with that establishes our technology as being very well suited in terms of the content, the sensitivity, the cost, and the turnaround time. The other big area, kind of the holy grail, of course, is in screening, right? And this is probably where the liquid biopsy notion becomes compelling, but also very difficult to pin down. And there are companies, very, very large investments going after the notion of being able to screen either healthy people or screen people with certain risk profiles to determine if there's a early cancer. And of course, the best way to prevent cancer is to detect it early. So that's a very compelling vision. Agena has some relevance too in our, some of our technologies that we're developing, but it's not a big focus for us. We're kind of in a more of a sensing mode with regards to screening opportunities, because we do see it as longer term before the content really coalesces into a set that's well known enough and that the clinical utility can be determined. So in summary, liquid biopsy is going to be a massive opportunity. We see very significant long-term opportunity for Agena. We're positioning in areas that we're particularly well suited, and so far we've established an excellent sort of framework, a foundation, I should say, for the performance of our system.
Good. This is a good start to the last block of our questions that will be about the future. So you talked about some possible opportunities. But can you comment a bit on how you see the future of the company? How many people are you now, and do you plan to grow, and is it necessary, and then to which extent?
Well, I certainly can tell you about the company now. We're about 180, going on 200 people. I think roughly 40 or more in research and development. About half of the company is based in San Diego, our headquarters. A little more than half, about 100 people, where we do all of our development, all of our manufacturing. Everything is made and controlled within the company here, and then of course all of our corporate offices, functions are in San Diego as well, and then we have offices in Europe, in Hamburg, Germany, and in Shanghai, China, and in Brisbane, in Australia. So that's the footprint of the company. We have sales, support, and service around the world. Direct offices in the areas that I mentioned, and then we do operate with a number of distributors and partners, including ILS in Russia, and many others around the world. So that's the current footprint. The way we think about growth is less about growing footprint or headcount, and more about growing business. We see sustainable, long-term, strong double-digit growth opportunities for mass array technology and our applications, and actually explosive growth opportunities in China, where we have now over 10 panels being developed by a range of partners that will get approvals over the next several years by the China FDA. That will really fuel explosive growth into the hospital networks, which are massive in China, and which we've really have very limited ability to sell into so far. But also around the world, we're very focused. It is still a fairly small, actually, commercial team, but we do focus on the bigger laboratories and the larger opportunities. But there are many more, and as we grow, we're very responsible, because we're also accountable to our investors and our profitability. But as we grow, we expect to continue to our revenue. We continue to grow our resources to accelerate that further, and that's how I see this as a growing business. We're in markets that are growing very well. We're in some applications with exceptional growth and even explosive opportunities, such as liquid biopsy. And as it's still a relatively small player, we expect to gain share as well. So there are many avenues for growth, and the hardest strategic questions are always the ones that the answer is no. Which things we're not going to do. We have many compelling opportunities, and we're constantly evaluating what the best next ones are to do. And it's a good problem to have, but right now I'm not going to give too many clues on the future, other than we have a very attractive set of options and opportunities for the company.
And geographically, what markets do you consider most promising? Well, except China that you mentioned already, and why?
Yeah, well, it's one of the things that I find really interesting about this business versus some others I've been in the life sciences. When you're approaching the research markets, it turns out actually that research is very similar all around the world, right? It's similar. It's really focused on the technology. Everybody's trying to publish. Most of the publications are in international forums, and it's an international endeavor. And it makes a one-size-fits-all approach. It's actually, from a business perspective, it's a lot easier, but maybe a little less interesting. And in diagnostics, when you get into both regulatory, reimbursement, and just structural differences, right? All of those are different, right? Obviously the reimbursement is determined by government, and as well as regulatory policies, and but then structurally, how laboratories are established. In the U.S. there are large commercial reference laboratories, and then there are many CLIA high complexity laboratories. In other countries it's more centralized, like I mentioned, the UK. it's all national, mostly national laboratories. Even more consolidated in France, but in Germany there are a lot of private laboratories, and and then China, which is completely different. Massive hospital system, massive independent laboratory system, but highly regulated by China FDA, and a lot of inside game that goes on there, where you need to have inside partners to operate effectively. So because of that, there's actually opportunities in different in each of these areas. I mean, the U.S. is still the largest market. China has the largest growth, but quite honestly, the U.S. is our largest business. We have great momentum. We are in many very high throughput and prestigious CLIA high complexity laboratories. We have a very excellent offering for testing the SARS-CoV-2 virus, and that's actively in play right now. And it's the approach of our kind of a tools-based and enabling-based approach is really well suited for that LDT market in the U.S., which is alive and well, and we'll continue to feed that. As I said, in China, it's more of a partnership strategy, and so there we've established partners. We continue to bring on more for different application areas, so we have a very diversified menu being developed by different partners who, and actually in all the areas of molecular diagnostics, where we have panels being developed in pharmacogenomics, as I mentioned, in oncology, including liquid biopsy, in a variety of inherited diseases, particularly for newborn screening, which is a very large opportunity in China, and then also in infectious disease. So, those, the U.S. and China are our biggest opportunities. This is obviously a European based interview, and in Europe also a very interesting dynamic in the business. Historically, and it's still the case, oncology has been our biggest business opportunity in Europe. Some of that is back to the structural, some of the large opportunities like pharmacogenomics or some inherited disease testing that has been present in the US has not been as significant in Europe, and now they're starting to grow now. But historically, oncology has been strong, and actually there's a lot of leadership from the scientific community in Europe, and so we're involved in several large consortia and many academic collaborations, proving out and comparing the performance of our technology relative to others, many ring trials and such, and publications that have come out of that. And so, we see that being a very strong springboard for the growth opportunity in oncology. And now we're seeing pharmacogenomics start to really take off in Europe. And so that's kind of the next kind of arrow, if you will, in the quiver for growth in Europe. And we're very well placed in all three of those major regions for growth, and each on its own kind of path based on the dynamics of those local environments.
My last question will be kind of visionary. What is your personal dream related to the company's work? If you could pick in the fantastic scenario, you want this or you want that, what would be your idea?
Yeah, look, my dream is that this technology, really becomes a standard, for targeted genetic testing, like the go-to solution for that, an iconic brand that everybody knows about, Agena mass array for targeted testing. I actually think it's the execution is the challenge and the opportunity, because the vision is that the world can see what we know, right? We know that we have absolutely the best midplex clinical testing platform on the planet, right? We've stacked up against everything that we've talked about, other alternatives to high-plex sequencing or low-plex PCR. In the midplex area, there's nothing that competes with mass array for these clinical applications. And we know that, right? So we know we're the best in the world. The challenges of a small company is building that awareness and establishing the momentum. And we're getting some real momentum now and increased awareness. And I see that just accelerating like a snowball, right? And so, you know, the vision that I have is that, and I'm confident that, you know, this is a technology we have that it's so well established now that we have, you know, more than just a beachhead. You know, we have a very established now position to grow from, and you know, that growth will fuel the company's reputation, and fundamentally it's about accessible healthcare, right? I think the real, you know, impact side of the dream is, and the whole reason why, you know, I'm even in this industry and why many are is to make a difference, right? To make, you know, meaningful diagnostic tests accessible, affordable, and impactful for patients' lives.
Thank you very much.
Thank you.